Hey everybody, I thought we could take a little bit of time to go over some of the work from last week. So I'd like to say that you are quite lucky I wasn't there. Otherwise this would have been a real quiz, but this time we'll just use it as some practice going forward into towards the end of the unit. And hopefully I will be back very soon. So if you have this cell division quiz, could you take it out? And if it was somehow collected, maybe just pause the video and then we'll go back. You can pass them back out to everybody and then we'll go over the answers. All right, so let's start with number 1. So this whole unit so far has really just been about cell division. We started with mitosis and what controls mitosis and how do cells know when to divide and when not to divide. And then we moved on to meiosis from there. So let's look at question number 1 here. Which one is true of the cell cycle? The correct answer is going to be A. So the cell cycle is controlled by 2 particular proteins. One is called cyclin, and it goes up and down as you move through the cell cycle, hence the name cyclin. And the other one's a cyclin-dependent kinase, which is also a protein, but it's an enzyme. And enzymes don't get used up and they don't get altered during the cell cycle, so they stay pretty consistent. So while these cyclins are going up and down, the kinases are staying the same. So when the cyclins are the right amount, they collide and combine with these kinases, and they push through the cell cycle. They really provide the energy to move through. Choice B is wrong simply just because that's what cancer cells don't do. They do not exhibit density-dependent inhibition, meaning that when it's dense, when it's crowded, they stop dividing. Cancer cells don't have that signal. So they pretty much will divide indefinitely if you give them all the nutrients that they need. Letter C is not quite right because there are both external and internal signals. So external might be something like, a cut on the skin will signal the cell cycle to repair or replace some cells in a particular area, but your body also has like naturally occurring moments when it knows to divide or signals that are produced, like for example, growth spurts that you go through. It's all predetermined in the DNA. So that would be an example of an internal signal. And letter D is not quite right simply because mitosis means division and interphase does not. So mitosis is dividing the nucleus. Interphase is just the rest of the cell's life when it is not actually dividing. Okay, on to question B. We talked a little bit about something called G0 or G0. And G0 is the time at which cells are at timeout. And that timeout can be permanent, but it can also be temporarily. So in the case of permanently arrested, meaning permanently stopped, in G0, these are going to be letter D. These are going to be your nerve cells. So your neurons, it also includes muscle cells. We talked about cells of the eye. These cells do not have the ability to regenerate. So whatever you have once you reach adulthood is what you have, and they never get replaced. And the $1 million evolutionary question is, how come humans got the short end of that stick there? There are other animals out there who do have the ability to regenerate tissues. They clearly have some genes that we don't. Very jealous. All right, number 3, a cell that passes through the G1 checkpoint, where is its probable destination? It's probably going to divide. So a cell that passes through G1 is most likely going to divide simply because G1 is the most important checkpoint. It checks for the most stuff. Checks for size, checks for room availability, checks for nutrients. And it kind of is like the first go-ahead, like, hey, cell, we probably aren't going to make more of you, so do you have everything you need? Okay. All right, let's move on to the S stage, where you'll copy your DNA. So it's kind of like the first gatekeeper. Okay, number 4, which one correctly matches the cell cycle with its description? The correct answer is weird here. It's letter D is the correct answer because it's thinking about G1 in a different way. So it's thinking, okay, mitosis has just occurred and we now have 2 little baby cells. Where do they go? They immediately enter G1. So G1 will immediately follow cell division. All the other ones are kind of mismatched. N is going to be the mitosis part. S is the copying of the DNA. And then G2 is the preparation for cell division, but you haven't actually divided anything at that time. Okay, number 5, during which checkpoint are cell size and the completion of DNA replication assessed. So DNA replication, remember, happens during the S phase, during interphase, and immediately following the S phase is going to be G2. So your correct answer here is letter B. The G2 phase is the one that checks cell size, but then also, really importantly, to make sure that all the DNA has been copied. Okay, number 6, during which checkpoint should all the chromosomes be attached to the spindle fibers? I feel like if I draw this picture one more time, I'm going to go insane. You're probably like, oh my God, again with this. So this is metaphase, remember, which is one of the most critical stages in mitosis. So this is our mitosis checkpoint. We need every chromosome to be attached to a spindle fiber because when they pull apart, we want every chromosome or every new cell to have that exact copy of a chromosome in it. Otherwise, you're going to have chromosomes with incorrect numbers, which is no good. Okay, number 7, which of the following is not a component of the MPF factor? So this is a protein. So this is really what we're trying to make when we combine the cyclin and the CDK together. This megaprotein is what drives you through the stages of the cell cycle. So we already know that cyclin and kinase are part of this. There's one more, and that's the phosphate, letter D. Phosphates provide energy. The source of that phosphate, it comes from 2 places. One is called ATP, and then one was called GTP, which we'll talk about later in the year. But the P, adenosine triphosphate and guanine triphosphate, those things, we can steal the phosphates off of them and use the energy in the bonds to power the process, to energize this MPF protein. It takes energy to go through cell division, it turns out. Okay, number 8. If there are 22 chromosomes in the nucleus of a toad skin cell, how many would there be in the egg? Okay, so this is a diploid haploid question. A skin cell is a somatic cell, which is basically any kind of body cell. And body cells are always what we call diploid. So this is a cell that has a double set of chromosomes, meaning 2 copies of every chromosome. An egg cell would be haploid. So haploid cells only have half the number of chromosomes. So it's super hard algebra here. But we would start with 22 and we would cut it in half if we were trying to find the haploid number. So letter C is your answer on that one. I hope you didn't need your calculator. This rule will apply with any organism I give you or any number I give you. The diploid number is always the full set, and the haploid number is always the half. Okay, next up, crossing over. So crossing over we looked at a little bit in the little video here. It's a really important stage in meiosis. It accounts for us creating variation in our offspring. So crossing over is important to create genetic recombination. It ensures that if we have multiple children or multiple offspring, that each offspring is going to be genetically slightly different. And in terms of evolution, that is good because that ensures, or at least gives us a little insurance, that at least one of our offspring should survive any changing environment. So meiosis is really important in sexual reproduction, and crossing over is really important in evolution. Okay, number 10, another diploid haploid question here. Which one describes the result of meiosis? So in meiosis, we have a cell that divides into 2 cells, and then each of those 2 cells divide again. If this were a human cell, we would start with 46. That would be our diploid number, and we would end with each one of these 4 having 23 chromosomes. So meiosis ends up with 4 cells, each one being haploid, or half the number of chromosomes. Okay, number 11, you are asked to culture, which means to grow, an unidentified sample of tissue. You notice the cells do not exhibit density-dependent inhibition. So what is the most likely source of this sample? Okay, so density-dependent inhibition is a normal cell. That's what normal cells do. If it's not displaying that, it's basically ignoring all signs of crowdedness, and it will continue to grow in the sample. So this is going to be an example of a cancer cell. And we talked a little bit earlier how there are probably multiple mutations that are dictating that inability to not be able to sense inhibition or not sense crowdedness. Okay, number 12. Tissue culture skin cells will stop dividing when which of the following occurs. So how do cells know when to shut it down? There's a couple of different ideas, but the best choice here is going to be letter B here. Letter B is all about the growth factors. So when cells run out of cyclin, let's say, or they stop producing cyclin for any reason, you're not going to be able to power the production of MPF, which means you're not going to be able to make more cells. One of the things that might signal the stopping of the producing of cyclins is crowdedness. So when it's too crowded and cells are pushing on each other, there's receptor proteins on the surface of the cell membranes that kind of signal, hey, shut it down, we've got enough cells. Okay, number 13 is a meiosis question. It talks about homologous pairs. So homologous pairs of chromosomes are two pairs of chromosomes. One would come from the mom, one would come from your dad. So we all have pretty much equal parts DNA from both parents, but during crossing over, they actually get together and they hug, they embrace very closely, and they swap. So the DNA on one side gets switched up. So my maternal chromosome isn't all mom anymore, and my paternal chromosome isn't all dad. It mixes up. So when these chromatids separate during meiosis, you're going to get variation in them. So that happens during prophase number 1, the very early stages of meiosis. That does not happen in mitosis. So prophase and mitosis, this does not happen. They're still homologous pairs, but they never come together in this way. Okay, and lastly, very important question here. Where do cells spend most of their time? The correct answer is interphase. So cells spend most of their time actually not dividing. If they did divide indefinitely and often, you would just continue to grow at an exponential rate. There was a lab that I was hoping you guys would get to do. I don't know if you did it. It was in your mitosis packet called Time for Mitosis. And it kind of plays a little bit into when we were drawing pictures and taking pictures of cells in various stages. Interphase you saw most often in every picture you took because that's where cells spend the most time. So if I took a camera and took a picture of a group of cells, most of them should be in interphase. The other phases, like the phases of cell division, as you move down this row here from prophase to metaphase to anaphase to telophase, the timing gets shorter and shorter. So like you might find a lot of metaphase, but you probably found very few telophase. And remember, some of you are lucky enough to find a cell in cytokinesis. And that is a rarity because the time it takes to actually divide that cytoplasm is so short that for a camera to actually capture it on film is quite rare. So we don't often see that. All right, so I hope that helps review some of the information that will hopefully pop up, will pop up on a quiz.